want to thank OAS for this invitation. I just want to give you a brief overview of our company, technology, products, and current stage of development. So Eyegenex is focusing on two technologies, gene therapy and peptide therapy for two leading eye diseases, glaucoma and diabetic retinopathy. Our leading product is the AAV2 gene therapy for glaucoma. And we have conducted a number of animal studies, followed by a study in non-human primates with this gene therapy. And we're in the process of filing a pre-IND submission in early part of next year, followed by IND enabling studies in about 6 months. And we also have a peptide therapy, which also we completed quite a few studies in animals, and we're in the process of doing a slow release formulation of the peptide. The diabetic retinopathy studies are very early stages in animal studies at this point, we hope to complete those studies probably in the next 6 months or so to take it forward into preclinical development. Our target population, as you know, glaucoma and diabetic retinopathy obviously are the leading cause of blindness in the world. And our target population for the gene therapy and peptide therapy are primary open angle glaucoma refractory to IOP lowering agents, which includes normal tension glaucoma. And obviously the diabetic retinopathy, we want to focus on patients with vision threatening disease. Mostly we want to focus with the gene therapy, which we are working on at this point for the diabetic retinopathy also. Our major interest is the gene therapy. Professor Nagaraj from the University of Colorado has done a lot of work in the heat shock proteins. And these are proteins that have anti-apoptotic properties. And what he found, and the literature also says that the HSPB1, lower levels of that protein is observed in glaucomatous retinas compared to non-glaucomatous retina. Also, there is evidence to indicate that a transient low level elevated expression of HSPB1 in RGC cells when they go through stressful response, which probably would lead to RGC cell death. So there is a very good evidence for the role of HSPB1 in the control of glaucoma. So at least plays an important role in the glaucoma process. So what he has done is he wanted to express the continuous presence of this particular protein so that you can control the apoptosis of the RGC cells during stress response due to increased ocular pressure. So he used AAV2, incorporated the gene for HSP1, and then also used a promoter that's specific for RGC cells. So he used this construct and conducted a number of studies in animals, in mice, mostly increasing the ocular pressure, using microbeads. And his early studies indicated that there is one intravitreal injection would lead to about 11% cell death in RGCs compared to the vehicle. There is almost like a 50% cell death in the RGC cells. So based on that observation, we conducted this recent study, whereas in this study, there were 3 groups. One is a age-matched control, and the second one is a vehicle control where the glaucoma was induced using microbeads, and another one is a same glaucoma model with intravitreal injection of the gene product with one intravitreal injection. So, and the animals are left for about a year. At the end of the year, they were, the study was terminated, and then you can see here on the picture, on the top, you can still see after a year, there is a significant expression of the protein after 1 year, one intravitreal injection. And also you can see at the bottom, there is significant also reduction in the middle where the vehicle-treated group, the RGC cells, compared to either the age-matched control or the one that had the gene injection. And those are also expressed in the bar graph below, where you can see a significant reduction in the RGC cells in the vehicle-treated group compared to the injected group. Whereas the age-matched control and the gene injected group had, almost a similar kind of RGC cell numbers. But more interestingly, if you look at the vision in these animals using, measuring the PA1 amplitude of the PER responses, on the extreme right is the group that has the gene injected. And you can see there's a significant improvement in the vision compared to the yellow bar, which is the glaucoma animals without the injection, just a vehicle injection. And on the other hand, what is interesting is in the naive animals, in fact, you saw a lesser vision compared to the gene injected animals, maybe suggesting that, with the age, there is some RGC reduction, whereas giving this protein continuously producing, maybe it reduces the death of those RGC cells. We don't know. We are planning on doing some more work in here. And here is another peptide, a small 19 amino acid peptide was taken from this heat shock protein, and he has done a lot of work in that, with that peptide also showing the similar kind of protection of the RGC cells in glaucoma animals. And we are in the process of doing a slow release depot formulation of the peptide so that we can inject once in 3 to 4 months as an intravitreal injection. We have a very well experienced management team that has startup experience from inception to the exit, including advisors here. So as a summary, we have 2 categories of products, gene therapy and peptide therapy, in 2 major indications, glaucoma and diabetic retinopathy. And we have one of our gene therapy product. We are going to take it into IND-enabling studies in about 6 months. And we're looking for investors and strategic partners to collaborate with us to take them for these products forward into clinical trials. Thank you very much.